Welcome everyone to this iteration of the IIT seminar series. So today we have Mike Fowler from Swansea University, who's going to be presenting to us about colored environmental noise and how that affects stability in populations. So without further ado, Mike, please, the floor is yours.
Thank you very much. Okay, so I'm going to start by thanking the organizer for the invitation. I've really enjoyed these seminars across the pandemic, and I'm very happy that they're continuing after the pandemic. I'm also really happy that they're recorded and being put up on YouTube, because that's helped me out a couple of times when I've not been able to make it at the live session. So thank you for the invitation, and thank you for doing such a great job managing this. Okay, I'm going to talk about colored environmental variation and stable and unstable population dynamics today. Let's see if I can make this move. There we go. But first, this is definitely the material I'm talking about today is all highly collaborative. So I'm going to thank past and present collaborators from all across the world, particularly in Europe. I spent a lot of time as a postdoc in Finland, working with a great group there. I've got some Swedish collaborators who are involved, and some are present today with the material that I'm presenting. I've got UK collaborators and PhD supervisors who have kind of given me the flavor for theoretical ecology originally. When I finished my undergrad as a zoologist, and then discovered the excitement of theoretical ecology after that, and it's held my interest since then, and it remains my favorite avenue to explore. Other collaborators in Ireland and from China and working in the UK or working in Ireland previously, and from France and the USA. So there's a whole great team of people I've really been lucky and enjoyed working with over the years, and they're all involved with the work that is being presented today. Okay, so I'll give some background definitions. First, colored processes this comes from a framework that's used to characterize random variation over time or space. And the term color comes from the analogy with visible light. What are the dominant frequencies? Are high frequencies dominating, or are low frequencies dominating? So in blue time series, high frequencies, rapid fluctuations tend to dominate. In red time series, longer wavelengths or slower changes tend to dominate across time or space. And in white processes, it's a purely random process where there's no dominant frequencies. Throughout this talk, I'm going to kind of interchangeably talk about stochasticity or noise, and it's usually in the context of environmental variability that I'm thinking about this. When it comes to colored noise, there's a couple of different ways that you can generate or analyze the noise, either with first order autoregressive or higher order autoregressive, if you want to get fancy, or 1/f kind of spectral synthesis or spectral exponent type methods for generating and analyzing it. The way I tend to think about it, the way I was introduced to this idea, is the autocorrelation. So if we think about what the current state of the environment is, or some other serial process across time or space, how does that relate to the future state of the environment, one time step ahead, usually? And if it's negatively autocorrelated, we're talking about blue noise. If there's no autocorrelation, it's a kind of uncorrelated temporal or spatial process. That's white noise. White doesn't show up well on a white background, so I've colored it black here. And then we've got positively autocorrelated noise is the red noise. Okay, some simple examples of environmental time series in this case, and the colors associated with them come from the North Atlantic Oscillation. This is the annual mean values across 180 or so years, and that tends to be basically a white process. The autocorrelation coefficient estimate is close to 0. The spectral exponent estimate is relatively close to 0 as well. We can do the same for monthly sea surface temperature, so a different spatial scale. And we tend to see these kind of seasonal fluctuations dominating the spectral exponent. So if you look at it monthly rather than annually, it tends to be a more reddened sort of process. And the final example here is how human interventions can really change the environment that different species will experience. So this comes from the Lokka Reservoir in northern Finland, where the previous management regime tended to change the water level slowly in the reservoir. It might have been more of a natural process, but then towards the end of the 70s into the 1980s, the change in management led to much more white types of water level dynamics. And that might have impacts on fish that breed in the shallows, for example, or the algal populations, zooplankton or phytoplankton. Both of them interacting with each other and so on. So these are kind of natural or semi-natural environmental processes that we can characterize by the color of those processes, how they change, in this case, over time, but you could be interested in spatial color as well. Back in the 70s, this sort of thinking was also applied by Roughgarden to population dynamics. So this is a kind of deterministic framework. We're moving away from the environmental variation or the stochastic component and thinking about the intrinsic component, and we can apply autocorrelation or spectral density approaches to population or deterministic population time series, and characterize them in the same way. Are they growing quickly, or are they growing slowly? What are the dominant frequencies in the dynamics that we see? How does biological parameters, intrinsic growth rate, for example, of the population, how does that influence the color of the dynamics that we see?
I'm so sorry to interrupt, but there was, I think, a pertinent and very quick question on the chat. What is the definition of alpha and beta?
Alpha, sorry, very good question. I've skipped over that. So alpha is the autocorrelation coefficient. So whether that's something between -1 and +1, going back to these images here, what's the slope of this line basically, or the correlation here? That's the alpha coefficient. I'm skipping merrily over the beta coefficient because that's a bit more in depth, but that's the spectral exponent. So that's when you can throw a lot of sine waves of different wavelengths together at different amplitudes. And I really don't want to get into this, I'm sorry, because I deliberately avoided including some images about that. But that's a different way of kind of characterizing the dominant frequencies in the time series. If you just fit, one long sine wave and several short sine waves, what would that be? Short wavelength sine waves together, you could ask which is the most dominant in those, or how those different wavelengths or frequencies appear relative to each other within these sort of random series. So beta is the spectral exponent there.
Thank you.
Good. Apologies for skipping over that. Right. What was I saying? So yeah, moving from the 70s into the 80s, there was a little bit more interest again in these ideas. John Steele was starting to look at again at more environmental variables. What's the characteristics of white noise variables and how they change over time? This is maybe a good example of the spectral exponent here, where we've got different frequencies. We decompose some time series into different frequencies along the x-axis, and then we ask what's the energy associated with each of these frequencies, sine waves of different frequencies here, for example. And then the slope of this line, if you fit a regression line through your data here, would give you the spectral exponent. Let me move on. So that was applying it to, well, in that case, geological records or other sorts of non-population records. And Stuart Pym and Andrew Redfern decided to apply it to population time series that were a combination of intrinsic and environmental processes. So these are all natural population time series. And they showed that with red noise series, we expect to see an increase in the standard deviation of the series. as the census period also increases. Whereas with a white noise time series, you tend to see it's a much flatter relationship. You don't really expect a big change in the standard deviation or the estimate of the standard deviation of the series as we increase the length of the time series. What are they looking at? Skylarks here. Swansea is actually an incredible place if you like skylarks. I can take my dog in the spring and the summer. I can take my dog out to walk across common land and listen to the skylarks screaming away at us. It's quite enjoyable. Right, moving on into the 90s and into the 2000s, John Halley was really influential in kind of popularizing this idea again with ecologists, bringing it back to the forefront of the discussion. He's got a great review paper in Tree, and then he continued with a few other papers kind of highlighting the importance of characterizing either environmental time series or ecological time series using these colored approaches. So this is a kind of brief literature review that's kind of piqued my interest in these topics. And the last one I think I'll mention just now is Vassar and Jonses. Their ecology paper was another really useful paper for me to get to know. And they went out and estimated a whole range of different environmental variables in the terrestrial and the marine environment, and asked what kind of color these were. They tended to be, well, they're whitish in the terrestrial environment. And again, the scale and the habitat that you're in will influence the sort of colors that you end up seeing. And the marine environment, they're much more reddened sorts of series. And again, thinking about the scale or the temporal scale, that can have a strong influence on the color, the exponents that you're estimating on these colored time series or on these time series. So I'll go back a second to thinking about the intrinsic dynamics. So these are deterministic systems. So there's no noise influencing the populations. These can be slowly growing, or they can respond slowly to a perturbation. That would be a sort of red dynamic here, or they could show rapid fluctuations that kind of dampen down to a steady state equilibrium as well. So but these are much faster dynamics. So they would be similar to the blue dynamics that we've seen in the other cases. I've simulated these with a simple single species Ricker function. It doesn't have any environmental fluctuations in it at the moment, but we'll add those in soon. So this is the sort of thing we can add blue intrinsic dynamics to blue, white, or red noise, or we could add red intrinsic dynamics to blue, white, or red noise, or we could add perfectly compensating dynamics to this as well, but they are maybe a special case that I'm going to skip over for the time being. Another couple of important definitions I want to make in the most of the results that I'm presenting here. I'm going to think about feasible communities. So all species will have positive equilibrium densities, and I'm also going to focus on asymptotic or locally stable dynamics. So in a discrete time system, that's defined as the dominant or all eigenvalues of the Jacobian matrix being less than 1. So the absolute values of all of these can be less than 1. There's other measures of stability like reactivity and resilience, but I'm going to focus on biomass stability, which is just the grand sum either of, or it's the variance associated with the sum of individual species population time series over time. So we get the ecosystem or the total community biomass, and we ask about the variability of that series. You can also find that by looking at the variance covariance matrix, taking the grand sum of the variance covariance matrix of each of these individual species. That tells you exactly the same thing. And this figure and the equations I've borrowed from Bart Hageman's preprint. I don't think that's been published yet. I checked it up again. It's on bioRxiv, but it's a nice summary of a lot of these concepts. So thinking about communities rather than single species, Tony Ives and collaborators published this, again, for me, a really influential paper in 1999, showing how you shouldn't expect to see anything other than a sort of positive biodiversity stability relationship. So this is the inverse of the variance we would see. Is it, or is it invariability? I can't remember just now. So essentially, as we increase the number of species in the system, the community level biomass is expected to decrease. And that's something like the insurance effect. So as some of the species respond positively to fluctuations in the environment, others will be responding negatively, and the more species you have, the more sort of compensation that you've got there in terms of their responses to environmental fluctuations. That's if they're all uncorrelated in their response to the environment. And you see a similar thing as you increase the correlation between species responses to the environment. So if they're all responding in very similar ways, then we kind of see a rather flat relationship. We still don't see a positive relationship. And this paper shows that we should only ever really see negative rather than positive relationships, or relatively flat relationships between community size and the variability that the total community biomass shows over time. There's a few more papers that built on that, and they culminated, in my opinion, in this Greenman and Benton paper in theoretical population biology. And it gives us a really simple looking recipe to calculate the variance covariance matrix, the population variance covariance matrix, I should say, accounting for the correlation structure in the environmental signal and accounting for the color of the environment. So that was an important step forward in this paper. So the simple white noise equation for the population variance covariance matrix, which is the way they've calculated this, it ends up, all the columns of the matrix end up being stacked on top of each other into a single vector. We're basing it on identity matrices and Kronecker products of Jacobian matrices, which I'm not going to go into the details of that, but you end up with one much larger matrix when you multiply these Jacobian matrices together. And again, we have this stacked environmental variance covariance matrix. So that's where our different correlation structures can feed in the environmental responses. And then in the appendix of this paper, they give the same sort of recipe, but we start to include environmental color into the functions here. And it works really beautifully in relatively simple systems. By relatively simple, they also include age and stage structure, or species interactions, and potentially spatial interactions. I haven't actually looked at that, but I imagine it works so well with the other cases. I imagine it should work quite nicely with that as well. So basically, what they're saying is, in that paper, is that when you add a blue environment to a red population dynamic, so a slowly growing population plus a rapidly growing or a rapidly changing environment, you'll tend to dampen out the effects of each of them. So you get a relatively small variability in that series. If you add a red environment to red population dynamics, you'll tend to get much larger variability in the series. And the same is true with blue plus blue. You get larger variability than blue plus red dynamics and environments. So we can either amplify or suppress the signals when they're added to each other. And that always just reminds me of a high school physics class. If you have a sine wave plus another sine wave of a similar wavelength, then you get constructive interference. You increase the size of the resulting wave. If you have a sine wave plus a sine wave with a very different frequency, then you get destructive interference. Or if they're out of phase, you'll get destructive interference. So that's the way that I kind of remind myself how this all works. And in a paper with Lasse Ruokalainen and other colleagues from Finland, we just checked that across different scales of biological organization. So it works very nicely. In single species, slow-growing populations will tend to show more variance in red environments than in blue environments. And the opposite is true for fast-growing populations. They'll tend to show higher fluctuations in blue environments than red environments. And we can scale up to a meta-population, and that general pattern holds. And we can scale it up to multi-species community dynamics, and again, the general pattern holds. So this seems to be a kind of a common finding with simple, relatively simple. We used kind of Ricker multi-species models or Ricker spatial models or single species Ricker models here. So I'm going to take another sort of sideways step into definitions again and talk about the asymptotic stability of this Ricker function, multi-species Ricker function. And what we're interested in again is I'm always dealing with feasible systems. So all population dynamics have all populations have an equilibrium greater than 0. I need to know the Jacobian matrix, which incorporates information about intrinsic growth rates and species interaction, strengths of species interaction. These tend to be competitive communities that I've looked at. And we're assuming that all eigenvalues in the system, and the dominant eigenvalue in particular, have an absolute value less than one. So if we plot the eigenvalues on the real and the imaginary axes that they can have, they're all within this circle with a radius of 1. And that's going to be important for some of the systems that I'm going to show shortly. This condition here, the absolute eigenvalue being less than 1, means that perturbations, a single perturbation will tend to die away, and the system will return to its kind of equilibrium attractor. And because these are stable point equilibria, that's relatively easy to keep a track of. This is another way of thinking about it. This is kind of May's classic 1974 paper, where he talks about stable equilibrium dynamics with lower growth rates, and they can go through a series of period doubling bifurcations and discrete time systems. So we get a 2 cycle to a 4 cycle to 8 cycles and 16 cycles until we end up with very bounded chaotic dynamics. And that happens when we have an absolute eigenvalue that's greater than 1 in these single species system. Actually, I think the next slide shows it. Yeah, we're falling off this side of the circle in the kind of cycling and complex dynamics system. So we're going towards negative eigenvalues, kind of less than -1 in this case. If in a multi-species system in particular, if we fall off this side of the axes, so we get the dominant eigenvalue is greater than 1, then we start to lose the coexistence. So species don't coexist. We tend to see some species going to extinction if the eigenvalues fall off this side rather than the negative side of the map here. Okay, so we can combine species with different intrinsic growth rates. So they can have kind of fast but damped oscillations, or they can be slowly growing to the equilibrium when there's no competition. So actually there's 3 species shown here with different intrinsic growth rates, but they're not interacting in this top left panel. Whereas if I switch on some competition between them in the lower panel, then they start to interact with each other, and we see that green species kind of damped oscillations disappear. There's a bit of an overshoot before it returns to the equilibrium. And that's the sort of systems that most people had been looking at around the time of the Ives and others paper in 1999, and for a bit after that as well. Another way we can look at that would be to ask what happens if we start to introduce species into these systems that do have higher intrinsic growth rates. So they might be showing cyclic or more complex dynamics, chaotic dynamics. So again, this top panel shows the situation with no interactions, no competition between the species. And in this case, the dominant eigenvalue would be greater than 1, or the absolute value of the dominant eigenvalue would be greater than 1. And you would have some fluctuations. And if we added all these populations together, then that would show some fluctuations across time. Whereas if we switch on competition between species here, we can bring this back to being a locally asymptotically stable situation. The competition starts to dampen out these chaotic fluctuations of the green species with the high intrinsic growth rate. So that seems to be an interesting option to relax the assumption that everyone in the community has got long-term stable equilibrium dynamics, and maybe some of them don't have that. And we can ask what happens as we start to vary population size, the number of species in the community, which is what I did in this now getting old paper from 2009 in Journal of Theoretical Biology. And I started off with just a simple diffuse competition, so all species were competing symmetrically with each other. And I started off with one species with a low intrinsic growth rate, and then started adding species that had higher intrinsic growth rates. And we can find out the eigenvalues for a two-species system, for example, should be relatively straightforward to find out. As soon as you go into a three species or more system, this equation starts to become pages and pages long, so it's much less appealing to look at. But we can also find a condition for the maximum growth rate of the second species that we add into this, and it has to be less than this, which we can plot out for two and more species actually. And above these lines, we find we're in the locally stable region, so they're coexisting because they're all symmetrically competing with each other, and they're not showing these kind of cyclic or more complex dynamics in this region above these curves. So a two-species system has a relatively smaller locally stable region than species with more than systems with more species. So as we increase the number of species, we're also increasing the strength of competition throughout the whole system, which allows us to dampen down higher intrinsic growth rates and dampen down these complex dynamics that we would see in the absence of competition. We can also look at that in asymmetric competition systems. So here we're assuming that the two species don't have the same strength of competition between them, and that can vary across every single species pair in the system. And we're asking, what's the probability if we randomize these alpha values and the intrinsic growth rates and draw them from some distribution indicated along this axis here with the maximum R value that we're drawing from. We can vary the community size along the other axis and ask what's the probability of finding a randomized community that's both feasible and locally stable. So it's showing stable point dynamics, and all species have got positive equilibrium densities. And we can see that, you know, as you have relatively high maximum intrinsic growth rates, we start to see these positive relationships between diversity and stability. If we're only selecting from species with low growth rates, then we only see that negative relationship there. So again, that was, for me, that was a kind of fun exercise to find out and start thinking about why would we only focus on this relatively limited range of dynamics, intrinsic dynamics? Why not allow species with faster growth rates or more complex dynamics into these communities to see what happens? And I started asking, is that a biologically reasonable position to hold? There was a lot of lab experiments in the 90s on the tribolium, the flower beetle, which were showing chaos, the onset of chaos, and you could switch between different types of complex dynamics, which there's a series of papers around that by Descharnais, I think, and Cushing and people like that. A really cool set of experiments and results. So at least in the lab, you can show it. And then in 2008, when I was playing around with these models, again, Tony Ives has shown a nice paper in a relatively simple system of midges in Iceland, I think. And they were talking about very high growth rates and switching between different dynamical states, which was cool. And then I've not read this paper yet, but it came across my desk very recently. People are still thinking and rethinking about the prevalence and relevance of chaos in ecology. So I'm glad to see these ideas are still popular. And I still enjoy thinking about them. So we go from the deterministic system back to adding noise to these systems. That's what I'm showing here. These 4 plots in the top right are the same as we saw before. So these are no noise, no environmental variability. And the examples I'm showing here are all based on white noise, because in these systems, colored noise didn't actually lead to any qualitative changes. But I think it's a nice result that we're going to come to shortly. So again, the left panels, we've got low intrinsic growth rates in the community. When we add some noise into it, we get some distribution of total community sizes or total population biomass sizes. On the right-hand panels, we've got the higher growth rates included. And if you're looking at a time series, there may be, you might guess that these are showing kind of more blue fluctuations, but it depends which one you're looking at. And when we simulate that over multiple replicates, we tend to find much higher, wider distributions of community biomass. I'm just going to check the time. Yeah, there we go. I'll move on. So this was the extension of the adding the cycling and chaotic species to these deterministic systems. So we're still dealing with locally asymptotically stable communities. We're drawing the noise from normal kind of white noise processes, but there could be some correlation structure in the noise that's the environmental signals are less than perfectly correlated, or the species responses to the environment are less than perfectly correlated. And then we just ask, what's the total biomass, the variance of the total biomass? And in this case, it's 1 over the coefficient of variation that we're considering. And when we've got perfectly uncorrelated environmental signals, I looked at this in the Ricker and also the Maynard, Smith and Slatkin model, another common, simple, relatively simple multi-species model we could use. We see, yeah, uncorrelated noise leads to positive diversity stability relationships. So these are fluctuating less than 12 species communities are fluctuating less than 2 species communities. However, as we increase the correlation in the environmental signals, we can finally start to see negative diversity stability relationships when you've got complex species with complex intrinsic dynamics in the system. So that was a new result for me. I don't think I'd seen that anywhere else before in these kind of simple competitive communities. And I was kind of pleased to find that. We can break it down into population responses, population level responses, so each individual population, and then the summed covariances. So again, thinking about the variance covariance matrix across the whole community. And this is just showing us that when the species are responding to the environmental variation in a highly correlated way, the summed covariance, the off-diagonal elements of the variance covariance matrix for the population, that's increasing faster than the population level variances are decreasing. So the scale, if you look at the scale here, it's going from 0.5 up to 0.2 or something, whereas the summed population variances are going down from about 0.1 to 0.3 or something. So this is a bigger change down here than that is. So that's why we end up with that negative relationship here. That's a kind of phenomenological explanation. We can ask about distributions of, what's this top one? This looks like distributions of RI values, so intrinsic growth rates, or we could ask about the distribution of eigenvalues for each community. And we can also ask about the way species are correlating and the introductions of species with high intrinsic growth rates into these systems. And there's fundamental changes, qualitative changes between systems where the correlation in environmental signals is 0 versus highly correlated. It's very hard to summarize how species are, how synchronized species are, or the correlation between different species dynamics when you've got more than two species. And this was one solution that we settled on in this paper, is just showing everybody's pairwise correlation, rather than trying to squash all of these into a single metric that maybe doesn't reflect the importance of individual or pairwise relationships. And it's full of beautiful colors. I also like that side of it. Right. So those were all competitive communities and not really worried about environmental correlation. I want to highlight this paper led by Martin Yang and colleagues when he was working in Dublin. I really enjoyed working on this with them. We showed this kind of typical variability increases with environmental autocorrelation. So these are slowly growing communities generally. And we found this really robust finding across 14 different types of food chains or food webs. That not only does variability increase, but other types of stability, like the extent of change or the recovery time. These are related to resilience and reactivity, I think. These are showing the typical kind of reddened dynamics plus reddened environments will lead to increases in variability or reductions in stability. What we also saw is that the predictability or the variability around each of these mean values also increases under reddened environments. And that's what we're showing in the right-hand panels here. So even though the variability maybe looks a bit lower in the recovery time, by the time you've scaled that by the mean value, we tend to see much less predictability. So higher coefficient of variation for this result here than this result here, even though it looks like there might be greater variance in the distribution of values here. So that was another really interesting result to find that it's not just the variance that increases, but it's the variance around that variance that's increasing. Okay, I'm going on to a more recent project, which I'll talk more about in a second, but this was, again, asking how relevant is this color information in terrestrial populations. And we started off by taking information from Jonas Knappe and Perry de Valpine, published a great paper in the 2011 in Proceedings of the Royal Society, where they statistically separated out deterministic population skeletons from the environmental signals. And then we decided to look at the environmental signal, along with David Gilliam led this, and Jonas and some other colleagues helped on it as well. So we would look and see, we maybe had a 10-year or 15-year time series of one species, and the original paper kind of matched up environmental time series from the Climate Research Unit database or the North Atlantic Oscillation or El Nino or something like that, to see, to kind of separate out the environmental signal from the deterministic signal. And we decided you could use that information to estimate the color along with an estimate of the uncertainty. So here we've got what looks like a reddened time series, but there's loads of uncertainty around that because we're dealing with a short time series as well. And we went back and pulled out the full 100-year length of the time series, of the environmental time series, and asked, if we maybe extrapolate beyond the length of the population series that we've got, does that change anything? What it tends to change is our certainty or uncertainty in the estimate of the color of the series. So over a longer time period, the same environmental series looks like slightly less reddened, but we're more confident that it's definitely not a white series. And you can do that with spectral exponents or with autocorrelation coefficients as well. Really restricted, well, there's a few examples here, four-spotted chaser and the eastern wood pewee, and they've got different lengths of the animal time series, and they're generally associated with higher uncertainty in the spectral exponent or the autocorrelation coefficient estimates. So we've got, we're really restricted by the length of the available time series. All of these came from the Global Population Dynamics database. So you start with about 5,000 time series, and then you whittle them down, get rid of all the duplicates, get rid of all the ones with missing values, get rid of all the awkward things that are changing the way they've measured the populations halfway through it and things like that. And we're left with about 250 animal time series. And then once we've thrown out all the duplicate environmental time series as well, we're left with about 170 environmental time series to play with. And we can ask which of these weather variables or climatic variables are associated with the population fluctuations, and when are they seasonal or are they annual types of things that are affecting them. And we generally find temperature, precipitation, frost, El Nino Southern Oscillation, and the North Atlantic Oscillation are the important ones. And there's relatively equal spreads across different seasons because different organisms are affected differently by different seasonal limitations, I suppose. And all the time series we've got come from birds, insects, and mammals. So this was an interesting paper to work on as well. The last part of this was really asking about that uncertainty. So when we estimate the color from these environmental series, how often does it differ statistically from white noise? And we can do that with the short time series that were matched to the population length. So that's everything is less than 50 years long. And we have relatively low confidence. There's a reasonable spread of bluish and reddish or pinkish colors in here, but very few of them are statistically different from white noise. And that dashed line tells us about the mean across all these different time series. And if we do it on the kind of extrapolated 100-year time series, we tend to see more of them are significantly different from white noise. And the larger proportion of those that are tend to be slightly reddened in terrestrial populations. So I think it's important to emphasize that you've got to go beyond point estimates and worry about how statistically different your estimates are from zero when you can. But I thought that was also sufficient justification to continue working with colored noise. And I'll rush through this, so we're not going on too long. I'm aware of the time, sorry about this. The last project I want to talk about is something we've kind of wrapped up just before COVID hit, which is delayed getting some of the results out there. But I'll introduce the team in a second. We've moved from natural populations back into the lab, and we're using a multi-trophic host parasitoid system based on the Protea interpunctella, which is a moth that's globally distributed, and it's a stored grain product pest species. So there's some applied relevance in studying these. It's got a natural parasitoid, Venturia canescens, which is a parasitoid wasp, which is a putative biocontrol agent. And we started asking, can we get some interesting interactions between environmental color and the cycles that the moth shows on its own, and the different sorts of cycles that it shows in the presence of the parasitoid. And we looked at things beyond population size, which I don't have time to talk about today, but I'm hoping we'll get some of this out in the next couple of years, looking at body size and fecundity traits, as well as some ecoevolutionary feedbacks. I forgot to mention that we've got it's relevant to study cycling populations. So we're going away from the locally asymptotically stable system. And there's estimates of 20 to 30% of natural populations show cycles, limit cycles. So it should be relevant to study this in the lab under controlled conditions. This is the team. Marianne was leading the experiments in Leeds under the supervision of Steve Sate. Ed and Laura were also up in Leeds as research technicians, and David and Chengwei were working with me here in Swansea on the mathematical models, working with stochastic delayed differential equations. So jumping from what would you say, out of the pan into the frying pan into the fire. The frying pan was a very comfortable place with those relatively simple Ricker models into the sorts of stochastic delayed differential equations. Is this going to play for me? I need to change this. So these are the actual true stars of the show. and it's all over with just like that. That's a parasitism event. So that's our attack rate parameter, is measuring how long the wasp takes to find and lay an egg inside this caterpillar, the moth caterpillar, and the egg will develop in there before exploding like a scene from the Alien films out of the mummified caterpillar. So it's a really cool system, biological system to work with. Let's move on. No, it's just replaying it. So yeah, we've got the Indian meal moth. It shows cyclic population dynamics over long periods. The Venturia canescens, the parasitoid, and Steve is leading these. He's worked in these systems for many years, maybe more than he'd like to admit. And the empirical systems consistently show these, high amplitude dynamics for both the host and the parasitoid. And they've showed it across multiple generations. So that's heading up to 2 years worth of data, 20 to 40 generations, something like that. And there's been solid theoretical foundation for this as well across a number of papers. So I'm really standing on the shoulders of giants by joining in with this. We repeated some of those kind of earlier constant environment experiments just to make sure there weren't any impacts of stock cultures on how these things behave. And we fitted our stochastic delay differential equation models to empirical time series, or compared them with empirical time series, and got a pretty satisfyingly high correlation when you average across the empirical replicates to try and get rid of some of the demographic stochasticity effects. Then we got a really high correlation between the simulation models and the empirical time series, at least for the hosts. Don't know if we did quite so well for the parasitoids, but it's a good start. We also ran kind of short-term experiments to look at the mechanistic impact of temperature on development. We know temperature has a very strong impact on the caterpillar stage. And they've got a kind of optimum temperature around 28 degrees in the lab. And that's very similar for the host as well as for the parasitoid. So we just did short-term experiments to see how their larval development would change across fixed temperatures. So we did one experiment at 22 degrees, one at 24, one at 26, and so on. So there's no kind of changing between temperatures and generations here. We also, to run the longer term experiments and models, we carefully matched the colored temperature series to make sure we were kind of using the same temperature values and try and match these up as closely as possible. Are you holding up a stick to me there, or is that a cat's tail? Yeah, try and get these matched up as carefully as possible. I'm going to skip over these slides because of the time. Here's a quick schematic of the stage-structured population model that we used. The important feature in this model, we've got eggs, larval, pupal, and adult stages for the host, and a larval and an adult stage for the parasitoid. And it's the larval stage that temperature is affecting in our simulations. And here's some equations for people who like those. The key point here is we could consider either a linear or a non-linear response to temperature in terms of the larval development times. And that gives us some competing models to consider in terms of linking between the simulations and the empirical results. And we've also got 3 pages of model parameters that we've taken from previous work as well to parameterize these models. So I'm not going to go through that. We can generate these really nicely behaved quasi-periodic cycles in the host and the parasitoid under deterministic conditions, and then we can switch on environmental variations, and these can be red, white, or blue as well, and ask what happens to the dynamics. We start to see increasing peaks, maybe decreasing or increasing troughs, and different sorts of increases for the host and the parasitoid. And it's maybe a bit easier to summarize these with phase space diagrams, where the we can look at it at constant temperatures. So we could have a cool environment, 22 degrees, and we get relatively low abundances for both the host and the parasitoid larvae. And we could also think about the maximum abundances under each of these temperature conditions. And what we see is a big increase in parasitoid densities as we go from low temperatures up to high temperatures. And that's both for the minimum and the maximum density on these cycles. Whereas the hosts show a big increase in their minimum density. So we're increasing from blue to red here along this direction for the hosts, but very, very little increase in, that should say maximum host density. Sorry, it says minimum. So that gives us an idea about the deterministic predictions, even though we're considering different temperatures. And then we can run it with stochastic temperature fluctuations based on a mean of 26 degrees and some standard deviation that takes us between 22 and 30 degrees. So within the limits that we've just seen here in these diagrams. And they seem to follow some sort of similar attractor across approximately 5 years or 10 years of simulating these. But if we superimpose that stochastic trajectory on top of the deterministic trajectories, we start to see some interesting emergent dynamics. We start to see, huge increases in the parasitoid densities. We see big increases, much bigger than expected, even under the best case, warmest case, maybe optimum temperature scenarios. So we get to see these surprising emergent dynamics. And there's interesting things going on in the troughs as well. This suggests we might get increases in pest outbreaks, even though we've got very high natural enemy densities, which might be related to temporal and phenological mismatches. And also the dynamics down at the troughs might suggest there's enhanced coexistence of both species here. So it's easy to get rid of one species and then potentially the other species if you're hanging around these low densities for any length of time. We tend to see these only in reddened environments. It's not quite so prevalent in white or in blue environments. I'm just showing a couple of examples here because we're still trying to get our heads around some of these results. And we can also think about the distributions and the kind of temporal structure of the development times. So that's another route into understanding what's going on here in red, white, and blue environments. These seem to differ. The distributions of them seem to be wider. There's a wider distribution of larval development times for the host and for the parasitoid under red than under blue or white environments. So this is a really useful key into what's going on, even though I've not fully understood this yet. Just to finish up here, I decided to ask if these kind of emergent dynamics happen in much simpler host parasitoid models. So I'm using examples from Kendall's 2001 paper. And again, it's a quasi-periodic host parasitoid thing. The deterministic cycle is shown in black. And we see much more kind of symmetric fluctuations around that cycle. I think this is with a red environmental variation around that. So we don't see those emergent kind of host and parasitoid outbreaks in this simpler model. So there's something interesting going on here. I'm looking forward to getting stuck into that again very soon. Okay, thank you for putting up with me. Conclusions are here. We tend to find in simple systems, colored environmental variation amplifies intrinsic biological dynamics of a similar color, and that holds across different scales of organization. When we start to introduce non-stable dynamics into systems, we can reverse kind of classical diversity stability patterns. And then when we completely exclude stable dynamics and we're only interested in cycling or non-stable, I suppose this is still a stable attractor in some respects, but non-stable point equilibrium dynamics, something else is going on that's interesting. I'm not sure if this is a kind of resonance effect, adding red fluctuations environments to red dynamics might just explain this, but I think there's something different going on because it doesn't happen in simpler host parasitoid models. And we'll be looking into the dynamics of the larval development time in particular to try and get a handle on that. There we go. Thank you all so much for your attention. Thanks again to the organizers for the invitation. This is a really beautiful photo taken by Ed, the research technician in the on the project. He's well worth a follow on Twitter, or maybe some other place if he's moving over there because Twitter's dying. But I'm going to stop there. Thanks again.
Thank you so much, Mike. So as usual, if you have any questions, just raise your hand in the participant list and I will call on you. Yes, Nadav, please go ahead.
You're on mute.
Sorry, I was muted. So thanks again for the very interesting talk. And there is something which I got confused when I looked at the literature on colored noise, which is what people exactly define as colored noise. And I mean, you may think about white noise as having different, whatever, you pick different environment every year, right, in an uncorrelated manner. Then you can take it in a correlated manner. So effectively, you pick new environment every 2 years or every 4 years, but the correlations are still exponentially decaying. There is a time scale above which you lose correlations. And there is another interpretation of red noise, which is that you have the decay of correlation is a power law. It's not an exponential decay.
Yeah.
So what kind of color of red noise you have mentioned here? Which of these is that noise?
So I've used autoregressive processes to generate the noise in most of these examples, and that would lead to the exponential decay. I think you can use spectral synthesis methods, which work very nicely with a single signal. And with two signals, if you're trying to get correlated signals. But as you start to build up from there, the tools that are available start to break down in their reliability for that kind of power law decay that you were mentioning. I think spectral synthesis generates more of a power law decay. I think.
Yeah, you can.
But when you're trying to get correlated responses across more than two species, or two spatial patches or something like that, or two life history stages, then the methods aren't that reliable. So that's why I've stuck with autoregressive methods for most of what I talked about today.
Thank you.
Yeah, thanks.
Thanks so much. Does anybody else have a question? Yes, Matthew, please go ahead.
I may have misunderstood this, but somewhere early on you were talking about measuring the total amount of variability as the sum of the elements of the covariance matrix. Is that right?
Yeah, that's correct.
So particularly with autoregressive models, I was wondering why that rather than something related to the generalized variance, because it kind of has an intuitive interpretation as a volume, and it also links up to lots of things like test criteria in multivariate ANOVA. So it seems like it's a thing that we know what it means, whereas for some of the elements, I don't really know what it means.
I mean, I've used that approach simply because that was the standard in the academic community, I think. I haven't thought about the general, what did you say, the general variance? What did you call it? Generalized variance. Right. It's basically just the determinant of the covariance matrix. But there's lots of related things that are used in multivariate ANOVA and that also crop up in things like vector autoregressive models. Yeah, I'm sure you could find out, you could apply it. I'm not entirely sure whether it gives you more mechanistic insight. So that's one of the drawbacks of the variance covariance matrix is if that's all you've got based on a natural set of coupled time series, that in itself isn't much use in a mechanistic sense, right? It's just a phenomenological description of what's going on. What Greenman and Benton do is give you the recipe to build up that matrix from the kind of mechanistic basis of your population and your environmental signals. would the generalized variance give you any more mechanistic insight, or you'd still have to define your model, I suppose?
You'd still have to define your model. I'll see if I can put some links in the chat to things where you can do stuff with it. It might take me a few minutes to do that.
Yeah, that'd be great. Thank you.
Thank you. Are there any other questions? Well, I have maybe one quick question. Can you get inspiration for the kinds of things you do from the disease literature, where this kind of forced dynamics with complex internal dynamics is a thing that they have been dealing with for a long time?
Certainly, and there's people who've worked with Steve in the past, Ottar Bjornstad, who's definitely done some of the disease dynamics. He's got a book out now. I think you can download it for free, which goes into different modeling approaches for disease dynamics. It looks pretty cool. And yeah, I think Brian Grenfell's done a bunch of cool stuff some years ago on disease dynamics. How a lot of that's on kind of human populations. So how you separate noise and intrinsic dynamics in a reliable way for human populations is probably something I don't want to touch with a 10-foot barge pole. But certainly it's an area of interesting exploration.
Actually, I just thought of something. Is the noise way too simple in those cases? Is it just a single sine wave as opposed to having a complex spectrum?
I mean, you could start with this single sine wave and just add more, right, of different frequencies and amplitudes, and build up to a more complex. That's exactly what the kind of spectral synthesis approaches do. You just start with a sine wave with a given frequency or wavelength, and then add some more of different frequencies and different amplitudes, and see what comes out. And you can force it to come out in certain ways if you want. But that would account for seasonal dynamics, I suppose. So that's useful for things like flu, and we'll find out if it's useful for COVID over the next few years and so on. There's been others they've looked at. I don't know if it's whooping cough or measles or these sorts of things. I'm not sure how seasonal those are, and how much you'd want a more kind of stochastic rather than sine type of environmental noise on top of that. But I'm sure there's lots of people who have done seasonal forcing with different amounts of stochasticity on top of that out there. I think Chris Klausmeyer's got his paper on the Floquet analysis. I think that it looks at some of that stuff as well. It's pretty cool.
Thank you. Are there any more questions? There are no more questions, then thank you again, Mike, and thank you all for coming. We will continue. Actually, Chris, do we continue next week or in 2 weeks?
It's next week.
Next week, thank you.
Double bill coming up.
Excellent. So next week, we will expect an email if you're signed up to the list, and we hope you to see you there. So thank you again for everyone's attention, and to Mike, and hope to see you soon.
Thanks a lot, everyone. Thanks for your patience.
Thank you so much. Take care. Bye-bye.
Bye.